Hi, I'm Stacy Lindborg. I'm Executive Vice President and Head of Global Clinical Research at Brainstorm Cell Therapeutics, and I'm pleased to be here today speaking to you on behalf of the company. I'd like to start by introducing you to Brainstorm's unique cell therapy product. It's an autologous product, which means that we use a patient's own cells to produce their product. As part of the manufacturing, cryopreservation allows us to create an off-the-shelf product for each patient, as one harvest of bone marrow creates several years of therapy. From the point of harvesting bone marrow, the first treatment is available in 19 days, with subsequent treatments being available in 7 days. Attributes that contribute to the product being consistent and reliable include things like there are no animal proteins or antibiotics, genetic modifications or viral vectors used. NurOwn is culture rescued, creating a product with a very high cell viability and consistent performance characteristics. And during the production of NurOwn, mesenchymal stem cells are expanded and induced to differentiate into MSC and TF cells, which are optimized to secrete neurotrophic factors. And lastly, NurOwn is manufactured with cell potency criteria, and all of these contribute to the reliable and consistent nature of this product. From a platform perspective, we've been conducting clinical studies for 10 years with animal studies in neurodegenerative disease animal models preceding this. And across these studies, we've observed consistent biology in vitro and in vivo data. And we confirmed the same neuroprotection, neurotrophic support, and immunomodulatory effects from earlier trials in phase 2 ALS population. Our knowledge of safety continues to grow across indications with more than 170 trial participants being treated to date. We're a publicly traded company that's capitalized on strengths of our R&D center in Israel by complementing the leadership team with experienced executives from pharmaceutical companies based in the US and Europe. We've taken the best cell therapy and we've applied this to neurodegenerative disease. This slide shows our growing development pipeline, and as you can see, we've leveraged our innovative cell-based platform and progressed clinical development across a set of neurodegenerative diseases. We're currently conducting clinical studies in 3 areas: ALS, progressive MS, and we'll soon be starting a trial in Alzheimer's disease. Our flagship program is in ALS, and the phase 3 trial is fully enrolled with top-line data expected to read out in the 4th quarter of 2020. We're proud that this was able to continue as planned through the current pandemic, and we hope that we'll soon be bringing positive news for people suffering from this horrible disease. In parallel, we have a phase 2 study going on in progressive MS, with top-line data also expected in Q4 of this year. And our Alzheimer's study is scheduled to start before the end of 2020. As I've referenced earlier, we've been conducting ALS clinical research for more than 10 years. Our earlier trials have been published in JAMA Neurology and Neurology and are easily accessible if you'd like to learn more about the evidence generated in these trials. Prior to phase 3, we completed a rigorously conducted phase 2 study that identified dosing, safety, and an important efficacy signal, which I will now take you through and then conclude with our phase 3 trial design and primary endpoint. Our phase 2 study was a double-blind trial with 3-to-1 randomization to NurOwn, or MSC and TF cells, and placebo. 48 trial participants total were randomized and had a 12-week run-in period, which is important in a disease like ALS that has such great heterogeneity in disease progression across patients. During this period, we had 4 collections of the ALS Functional Rating Scale, which allows us to understand the rate of ALS decline for each participant prior to treatment. And this allows us more precision in our estimates of treatment, the treatment effect in the treated period. In this trial, participants received 1 treatment and then were followed for 24 to 26 weeks. And the study was primarily a safety and tolerability study, although we collected efficacy data and CSF biomarker data. Fast progressor criteria and clinical response criteria was also established a priori. We can see from this slide that a single dose of NurOwn slowed the rate of progression by improving the ALS-FRS-R average rate of decline post-treatment compared to pre-treatment. The difference on this slide between panel A and panel B are the trial participants included in the analysis. Panel A is all trial participants, and panel B are the subset of rapid progressors. And as it was noted on the previous slide, fast progressors were defined as trial participants with a greater than 2-point decrease in that 3-month lead-in period. So as we look to panel B, we can see there's a more pronounced treatment effect as measured by the change in the rate of ALS decline post-treatment to pre-treatment in trial participants whose pre-treatment ALS decline was more rapid. Another way of observing the treatment response is to leverage a clinical response criteria. A participant is labeled a responder if they have a 1.5 or greater points per month improvement in their ALS-FRS-R slope post-treatment compared to pre-treatment. We observe a treatment effect in the left panel, which again is reporting all trial participants enrolled in the trial. So we observe a larger treatment effect in all trial participants that were treated with NurOwn, and a larger percentage of participants achieving the clinical response versus placebo at all time periods. In panel B, again, we observe a more pronounced treatment effect in the subgroup of rapid progressors, with some points reaching statistical significance compared to placebo. When we look at our biomarker data, we observe a few important things. The cells are delivering their cargo and are secreting increased levels of neurotrophic factors, while changes of pre and post treatment for placebo-treated participants don't change. You can see this, the blue in the slide represents pre-treatment and green represents post-treatment. If we look at inflammation and inflammatory markers, we also see a significant reduction in these, as illustrated by MCP1, which is an inflammatory mediator. And again, there's a reduction of MCP1, in fact, as you can see on this graph, by 40% in NurOwn-treated participants, and there's no change in placebo-treated patients. The treatment difference is significant in this analysis. And for both the neurotrophic factors as well as inflammatory markers, I don't have the time to walk you through more data, but we see similar movements in all of the markers that we collected. And I also can't take you through additional biomarker data, but I do want to remind you that of the consistency of the read-through in the effect that we've observed in NurOwn, in not only being delivering a neuroprotective effect, increased neurotrophic support, and immunomodulatory effects in NurOwn. This really does create a foundation to launch our clinical research of this technology platform, this cell-based technology platform, to many different neurodegenerative diseases. So I'd like to round out my time by taking you through the phase 3 trial design and then illustrating the primary endpoint. So what can you expect from this phase 3 trial design? The features are, there are a lot of features that are very similar to our phase 2 trial design. You can see the 12-week run-in period, the length of treatment of 28 weeks, also very similar, and a continued emphasis on biomarker data, of course, along with clinical endpoints. We are collecting 7 CSF samples in all trial participants across the study. The main differences which you'll observe in this trial, we're choosing to focus on fast progressors, as well as the number of treatments that are given across this treatment period is increased. In phase 3, we have 3 treatments that are given 2 months apart, and in phase 2, we only had 1 treatment. Our primary endpoint is a responder analysis that is assessing the rate of decline post-treatment compared to pre-treatment, similar to the analyses that I've already shown you from phase 2. And for our secondary endpoints, we have outlined a set of key endpoints which have been outlined in our SAP and have the appropriate control of type 1 error rate. These endpoints include looking at change in the ALS-FRS-R from baseline, the combined analysis of function and survival, which is often called CAFS. We'll analyze additional endpoints such as slow vital capacity, trach-free survival, as well as overall survival, in addition to the CSF biomarkers. And of course, safety remains paramount in all of our trials. So I'd like to leave you with an illustration of our primary endpoint, which I hope helps you understand the importance of this measure and the insights that we can glean from it. This graph illustrates two individual trial participants and their ALS-FRS-R scores over time. The placebo patient illustrated on this slide is in red and is at the top. And you can see the red triangles highlight the rate of decline, the actual data points, pretreatment, as well as the line that is fitted. And you can see the rate of decline, or the slope pretreatment, for this trial participant was negative 1.16, which is slightly over the 1 point per month loss on the ALS-FRS-R, which is the median progression rate estimated in the literature across the ALS population. Post-treatment, you can see for this participant is really quite similar to their pre-treatment rate of decline. Again, you can see the individual data points, the circles that are on the plot, and the estimate of the rate of decline or slope, which is negative 1.45. Comparing the pre-treatment slope and the post-treatment slope, there's a difference of negative 0.29, and compared to the criteria is clearly not a responder. The graph at the bottom of the slide and with the blue dots and lines represent a neuron-treated participant. And you can see from the pretreatment period that this participant had a rate of decline, or a slope, of negative 2.11 pretreatment. The dotted line is very important on this graph because it actually highlights the expected trajectory for this individual from their pre-treatment rate of decline had they not actually received treatment. The blue dots highlight the ALS-FRS-R scores at each of the time point measured. And then you can see that the post-treatment rate of decline, or slope, is estimated to be negative 0.21. So we see a stabilization of ALS decline in points per month in this individual, with a difference between the post-treatment slope and the pre-treatment slope of 1.89. So clearly larger than the 1.25 criteria defined in phase 3 as a responder, therefore making this participant's trajectory a responder, a clinical response. Then to complete the analysis for the primary endpoint, the response data is analyzed using a statistical model with important disease covariates, which are well established in the literature. So I'd like to thank you for your attention, and we look forward to sharing results with you from our trials as they complete in the near future.